All right. So welcome everybody to the Nerve series. Thank you for being here. Today, really excited that we have Dr. Kara Leyzac, who's an associate professor in the Department of Otolaryngology here at MUSC, where she also serves as the director of the Cochlear Implant program. She completed both her doctorate of audiology and her PhD at the University of Maryland, College Park, where she studied interactions between aging and cochlear implants in adults. And then she went on to join the faculty at the University of Michigan, where she was part of their cochlear implant program for several years. And she did some exciting research there, among other topics, with Susan Shore's group looking at neuroscience of tinnitus. Prior to coming here to MUSC, where she leads the CI program. She's done really interesting research on several different topics, and one of those being looking at relationships between underlying health of the auditory nerve, neural processing, and how people fare with cochlear implants. And today she'll be giving us a talk entitled Neural Processing of Speech in Cochlear Implant Users. And thank you so much for taking the time to be with us today, Dr. Leyzac. And without further ado, I'll hand it over to you.
Okay, I appreciate it. All right, so that's the title of my talk. Very exciting. Hold on, let me get to my slides here. Sorry. Okay. All right, so just to keep everybody on time, I'm going to go ahead and start here. Everybody here is familiar enough with cochlear implants. I don't need the obligatory slide sort of explaining what they are. And you all should sort of be familiar enough as well that, we all know that patients are highly successful with cochlear implants. But even those who are most successful still share some feelings like those shown on the slide here. Oftentimes patients will say, man, I love my implant, but, and then they'll list 10 things that they hate about it. That's pretty normal. So the point of that is that, even though they're super successful and we've come a long way with cochlear implants, there's still a lot more that we can do and a lot more that we don't understand about how they work and what factors we can actually modify to improve performance in our patients. So before I get into sort of what this talk is really going to focus on, which is sort of neural health, broadly speaking, I'd like to acknowledge first that there's many, many other factors that play a role in how somebody actually hears with their implant. So these are all, again, should be familiar with this audience here, but cognition, electrode location, how many hours they use the device, and age, all of these things will affect how somebody actually hears. And it's not to say that they're not important, it's just not the thing we're going to be focusing on for the talk. So when we talk about sort of neural processing, the first part of the talk is going to spend a lot of time talking about the auditory nerve itself. We're still sort of exploring this in a way, and we're certainly, not the only group that's looking at this. There's been a lot of attention on this in the past 10 years, and I'll get into that a little bit. And then since I've come to MUSC, a lot of what we've done is really sort of looked at the sort of the top-down cortical encoding of that information as well. I have a lot of information about that as well that unfortunately, I can't share with you today. We're still sort of digging through that data, but I'll share with you a little bit about sort of the patterns that we're seeing, specifically with temporal encoding at the level of the auditory cortex with these patients. So we all know that this is what a cochlear implant looks like. This is the electrode array. And the thing we're going to be focusing on, like I said, for the first half of the talk, is sort of the condition of the spiral ganglion cells, which are adjacent to each of these electrode contacts. This would be sort of a perfect situation in which there's very robust, healthy population of fibers that remain. And it doesn't always look like that. And so we're sort of trying to explore exactly what that looks like in these patients. How do we actually measure it? And then what do we do about it? How can we use that information to understand CI outcomes a little bit more? So when I think of spiral ganglion density, this is the best way to describe it. Probably don't need to do that for this audience, but it's always a fun slide to throw in. The density is really just that. So in a highly packed cochlea and the one shown on the left, that's just a cross section of modalis in animal model. You can see that there's a bunch of cell bodies that are packed very tightly close together, very similar to the beach that's shown on top of it. And then in some other animals that are deafened a different way, you can see that there is the density of those fibers is much reduced, akin to the beach that's on top of it that's quite empty, right? And so we can, we don't exactly know always what this looks like in our patients, but we can imagine based on either etiology of hearing loss, which I'll get into, or sometimes just duration of deafness, that the number of surviving cells looks very different across our patients, or perhaps even in different spots within the cochlea going from base to apex, similar to the animal histology slides that are shown on the bottom of the screen. So we're going to spend a lot at first talking about just the number of neurons that are surviving and how that actually affects speech understanding and how we can use that information to perhaps modify programming. So I would say up until maybe 10 years ago, that there wasn't a really good understanding of the relationship between the total number of cells that remain and how somebody actually hears with their implant. And I would also argue that a lot of people in the field, including myself, kind of thought, it probably doesn't matter that much, right? That you put the implant in and the brain sort of takes over and it figures it out. And a lot of those assumptions, I think, were based on the data that we had available at the time, which was a lot from these postmortem temporal bone studies. And this is just a sampling of the studies that existed at the time. And you can see very clearly here that there's a big discrepancy between these two figures. So you have the figure on the left basically showing that there's no relationship at all with the spiral ganglion cell count taken postmortem, and then the MUSC word score that was taken when the individual was still living, whereas the figure on the right shows this nice, beautiful relationship. And so there was a lot of conflicting information, but it was really these temporal bone studies that was the only thing that we had to go by in order to understand this relationship a little bit more. But yet there was all this existing evidence that sort of hinted at this relationship. And I think anecdotally, we've seen this in the clinic sometimes too, that certain etiologies of hearing loss have different outcomes. And so there was some additional postmortem studies that actually looked at how different etiologies affect the number of surviving cells in the cochlea. And so you can see on the far left, etiologies such as viral labyrinthitis, typically result in a significant reduction of the cell bodies remaining, and etiologies such as idiopathic sudden hearing loss. Some of those individuals are very similar to the amount that's left in somebody with normal hearing. And so we see that this can definitely vary across different ears, and it is somewhat dependent on the etiology involved. Taking that a little bit of a step further, several years later, there were some genetic studies that were done. This is the group out of Iowa. And I want to draw your attention really to, let me get my fancy laser pointer out maybe. Okay, so if we look at these two scatter plots here, this is, individuals that had sort of a known genetic component to their hearing loss that was really affected more of the neural substrate. And these individuals, it was more the sensory components or sensory organs. And you can see here that those that just have a sensory hearing loss really outperform those that have more of a neural etiology to their hearing loss. And then there's others here too, like otosclerosis, usually doesn't involve the cochlea significantly. Those also tend to be high performers. And so we saw these trends that sort of perhaps led us to think that there is a difference between the number of neurons surviving, or at least, what affects the neural population and outcomes. But again, there wasn't really this direct evidence to support this necessarily. One more slide about this, you look at sort of these extremes for genetic causes of hearing loss, congenital causes of hearing loss. And, those with connexity-related deafness tend to really do well with their implants. And they also, if you think about the sort of connexin-related deafness, all of the structures are there. They're just not all working together like they should. But normally these individuals, if you look at, histology in both the animal model or postmortem temporal bone studies, they tend to have for the most part, very well defined and sort of these high spiral ganglion nerve counts left. Contrary to that, we have children with CND. And we know that their nerves affected and they tend to do quite poor, most of them, or it's just variable. And same with ANSD as well. You have these more variable outcomes. And so again, we sort of see these themes that tell us that maybe the condition of the nerve is important, but this is about all we know for several years at this point. So we know that maybe neural health matters, but how do we actually assess it when patients are still alive? How do we assess it in humans? And it's important to understand that and to develop these neural health measures in order to better understand not only the relationship, but then if we're going to use these findings to actually improve how people hear with their cochlear implant, we have to have a way to effectively measure that while they're still alive, right? So most of my time at the University of Michigan was spent doing a lot of this. So I worked at in a lab at Kresge. We worked with both animal and human models and really sort of developing these measures based in the animal cochlear implanted guinea pig model, and then trying to apply them in humans as well and figure out how we can do that a little bit more. So I'm going to talk about that next. So there are many, many measures that have since been developed. So we started this work around 2016-ish, and we started using the electrically evoked compound action potential, or the ECAP measure, in order to use this as some sort of estimate of nerve health or the number of remaining neurons. And this is pretty much what we're going to focus on today. But there are a number of other measures, which I'll also at least mention for those who are interested in it. But today we're going to sort of focus on this potential, which really just consists of an N1P2 response. They are super easy to measure. It's one of the reasons why we did it. They don't take long to measure. They are typically used a lot in the clinical setting, just not in the way that we're going to talk about today. And I'll talk about that a little bit more. But very briefly, when we use them in the clinical setting, all we're getting is information about threshold, and that's it. The threshold measures don't really tell us anything about the way that nerve is performing at all. It doesn't tell us at all about how many neurons are potentially there. I won't get into it too much for this talk, but usually the threshold measures that we measure clinically actually correlate best with medial lateral distance, so how far the electrode is from the medialis, and that's about it. So you have to really move towards super threshold measures in order to estimate anything about the condition of the cochlea. And so the measures that we looked at, and I'll explain this a little bit more, but the measures that we looked at and specifically were these super threshold measures, the amplitude growth function, where if you increase the current level, that response, the N1P2 response, grows bigger and bigger and bigger. And if you do it enough times, you can get this lovely growth pattern. And there's two different metrics that we typically looked at for this. And one was this AGF slope, so this linear slope measure, and the other one was the peak amplitude. They are different, but for the purpose of this talk, they are similar enough that I'll use them sort of interchangeably. We know, and I'll get to this too, based on the animal model, that the peak amplitude and the slope kind of describe the same thing, and you can use them either one really. So what we found by looking at a bunch of were deafened using various deafening treatments, is that when we actually look at the growth of that ECAP slope function, and then we look at the number of cells that are remaining, SG cells that are remaining in the cochlea, that we find that there's nice positive relationship in the animal model. So the greater the number of neurons that are remaining, the higher, the steeper that slope is. We actually see the same pattern with a peak amplitude as well. So like I said, oftentimes those two metrics can be interchangeable. But what this really told us is that here's a measure that we can do in animals, we can do it in humans, and we think it tells us something about how many neurons are left in the cochlea. It's not a direct measure, it's more of a relative measure. And so when we look at that in humans, it does tend to look something like this. So this is kind of messy, but it's not like we can do the ECAP measures and say there's 50 neurons left. It's more sort of a relative measure. Where we can say, okay, the density of neurons is better in the medial electrodes than it is in the base, or vice versa. When we started to look at applying these measures, though, and really looking at these ECAP measures and these super threshold ECAP measures in humans, what we actually found when we compared it to some postoperative 3D imaging CT scans is that I guess it's not too big of a surprise, but these ECAP measures, the super threshold measures specifically, can still be very strongly influenced by the location in the cochlea. And so usually the closer the electrode sits to the medialis or that neural structure, the bigger the response is. The further away it sits, the smaller the amplitude gets, and the shallower the slope gets. And so this is sort of a confounding issue when we're talking about developing these measures that we can potentially exclusively used in humans, we don't always have CT measures available in adult or in humans, right? Sometimes we do, but most of the time we don't at a lot of clinics. And so we started to explore sort of these other variants of ECAP measures that were not necessarily as affected by electrode location. I don't have it on here, but just so you know, because I get asked this a lot as well, is scalar location actually doesn't influence anything. So when you sort of factor in scalar location, it doesn't significantly affect super threshold ECAP measures such as amplitude or slope or anything like that. So anyway, like I said, we started to sort of think of measures that we could use that still reflect the number of surviving neurons, but are not so much affected by where the electrode actually sits in the cochlea and how close it is to the medialis. And so we started looking at something called the IPG effect or the interphase gap effect. And the interphase gap is simply the duration of time between the leading cathodic phase and the following anodic phase. And so it's set by default at various durations with different manufacturers. But one of the things that happens, and I'll show you some data, is that if you keep everything else constant about the shape of the pulse itself, and you just increase the interphase gap, it actually induces excitability of the fibers. And so it will increase the amplitude if you increase the interphase gap, and it also increases the slope as well. But because you're just keeping the electrode location constant, it actually doesn't, it is not influenced by the location of the electrode and the medial lateral distance. And so here, some follow-up work in guinea pigs, basically very similar to the previous graph that I showed you, except here we're looking at sort of this interphase gap effect, or how much the peak amplitude changes or how much the slope changes. Again, it's very similar to this, and how many neurons are surviving. Okay, so the whole point of this is that we're really working to develop these measures that can be kind of easily applied in humans, that are irrespective of electrode location. They just really sort of focus in on the relative density of remaining neurons moving from base to apex. Okay, so what? So we found out that ECAP is a non-invasive estimate of SGN density in animals, and we also know that ECAPs can be easily measured in humans. We do it all the time, but why does this really matter? So we developed this measure, but does it really matter? Now we can sort of get back to the original question and answer, does residual neural health, or really does the number of remaining neurons actually influence how somebody hears with a cochlear implant? And so when we did the first study, this was a number of years ago now, we really wanted to try to strictly control it as much as possible. And so we chose 10 bilaterally implanted listeners. We measured these super threshold ECAP measures. The IPG effect for peak amplitude is the response that I'm showing you, or the measure that I'm showing you all here. And we measured that in the right and the left ears of these bilaterally implanted adults. And then we also measured speech recognition in each ear as well. And I'll get to that on the next slide. The thing that you'll notice here is that, in some individuals like this one, the IPG effect for the ECAP peak amplitude is very different across the two ears. And in other patients like this one, it's very similar. But that's just typically what we see with this. But what we really wanted to know is, does that at all relate to speech understanding, right? So the hypothesis here would be that if you have a larger IPG effect, that means that there is a more robust population of SG cells in that ear or that side. And so we would predict then that speech understanding would be more favorable in that side. So right ear, left ear, left ear. And we chose to do this in bilateral implanted patients because it does help to control a little bit, at least for all the other variables that we know affect how people hear with their implant, like cognition and all those things. So you can see here that when we do it in this very strictly controlled way, we do get this lovely relationship. Basically, these individuals up here have better speech understanding in the left ear, and they also have better neural population based on the ECAP measure. And then in the right ear, it looks like that as well. So we have some more, I'd say, more direct evidence, not just the postmortem temporal bone studies, but sort of mounting evidence collected when the patient was still living, that say that yes, at least to some extent, the condition of the nerve and really the number of fibers that are remaining affect how somebody actually hears with their implant. Okay. Recently, we've actually started exploring some other measures, and I don't have, unfortunately, I can't share a lot about this, but it's super interesting. So we're breaking down the ECAP measure even a little bit more, and we're looking at things that do not necessarily just reflect the number of neurons that are remaining, but sort of how those neurons are firing, and whether they're firing synchronously or whether there's some desynchrony there. And so we call this is just modeled after stuff that's really well known in patients with normal hearing, and particularly in an aging population. So I'm not going to touch on the aging thing so much in this talk, but know that aging effects are prevalent throughout almost everything I'm going to talk about today. I'm just ignoring them for the purpose of this, but this really comes from some aging studies in normal hearing. Um, but there is a way that you can sort of capture in normal hearing population, when you look at just the acoustically evoked compound action potential, ways that you can look at synchrony of the way those neurons are firing. And so we've started to try to look at that, some analogous measures in cochlear implanted patients as well. So the whole point of this measure, it's very simple, but what we think it actually tells us is about sort of the synchrony of the neurons. And what we would expect then is that if this firing pattern is very desynchronous, then it's not always going to look like this initial firing pattern, right? It fires a second time, it's going to be, a little delayed, it's going to be a little bit sooner. And what you're going to see here is a lot of jitter in the response. And when you average all those individual sweeps out, what that ends up being is really a longer N1 P2 peak latency value. So if everything's firing synchronously, things are going to be shorter in duration. And if things are firing very asynchronously, then the overall latency is going to be delayed. Okay. What's interesting is if we actually look at this N1 P2 latency measure, and this would be longer to the right of the graph and shorter here, that the more synchronous those neurons are firing, the better speech understanding and noise that they have. And the poorer those neurons are firing, or the more synchrony dysynchrony that's present in that neural population, those individuals tend to perform poor. Okay, so there's a lot more that can be said about this as well. The interesting thing about this is that if you look at the other ECAP measures that I've shared with you so far, the amplitude and the slope, which we know from the animal model, tell us about the number of fibers that are remaining. Those measures are not correlated at all with these latency measures. So it seems that just because, you could have a very robust population of fibers, but if they're not firing synchronously with one another, then you might not have great speech outcomes. And vice versa. So we're still getting into this a little bit more. It's pretty new, and we're looking at some other measures of synchrony as well. But the more we look at this, we actually think a lot of it is more due to synchrony than just the number of fibers that are remaining. Anyway, but that's relatively new stuff. Okay, so also getting back to, I said that I would mention some other neural health measures that I'm not going to talk about here. Some of the stuff I've looked at, some of the stuff I have not, but just to acknowledge that it definitely exists. Like I said, 10 years ago, nobody was looking at this stuff. The only evidence we had was postmortem temporal bone studies to help explain this relationship. Now there's 1,000 measures out there, and lots of people trying to develop these measures of neural health to capture this in patients. And so there's psychophysical measures, there's electrophysiologic measures. I'm sure everybody in this group is familiar, for example, with the intraoperative ECOG measure and the cochlear microphonic, and evidence to show that that relates to how somebody hears with their cochlear implant. But this exists, and there's a whole lot of literature that exists now that didn't 10 years ago. And if anybody's interested, you can look at it, but I'm not going to talk about it here today. So getting back to the initial ECAP measure that we talked about, looking at the amplitude and the slope response, which again tells us about the density of the remaining neurons, how do we actually use those results at all to help improve outcomes in patients? So when we talk about cochlear implant outcomes and factors that influence those outcomes, a lot of those factors are really non-modifiable. You can't, sometimes they're kind of modifiable, but for example, you can't sometimes change the age at which a patient walks into the clinic and decides they want an implant, right? You can't change etiology of hearing loss. But potentially there's things that I can be doing as an audiologist to do my job much better, to help program the device and maximize speech understanding for each listener that we have no idea about now. And I would argue as an audiologist that when somebody's having trouble with something, there's very few things that we can actually manipulate. We just assume that everything works well, the default parameters work, and that's about it, right? But there's really not a lot that we can do to modify things. So if we can find something, right, that's wonderful. So that's what we kind of wanted to delve into next here. So what we decided to do was something we call a site selection approach. It's been used by other people. It's sort of similar to, doing a CT scan and picking the electrodes that are further away and deactivating those. All you're doing is you're saying, okay, we have this metric that we're using to select which electrodes we want to activate in the clinical map or which ones we want to turn off. So if we're looking at the example here, you can see that this electrode is adjacent to nothing. There's really no neurons there or no peripheral process there anyway. It looks like there might be a cell body, but you would turn that off. Same thing with that one, same thing with that one. So the question is if we systematically turn these electrodes off based on the ECAP measures that we think tell us about the density of neurons remaining, do they actually hear better? Does that help? Okay, so this is a study that we also did a handful of years ago. We recruited 18 adult postlingually deafened patients. We measured the ECAP growth function on each electrode. We used the IPG effect for the peak amplitude. And then we systematically turned on or off electrodes depending on the ECAP results, right? So we did this sort of sectioned through the cochlea, so we didn't warp the tonotopic mapping, because that wouldn't have been good either. So within each section, we identified sort of like the best and the worst ECAPs and turned those off to create 3 different maps. So for one map, the 5 electrodes with the highest IPG effect were removed. Next one, 5 electrodes with the lowest were removed. And then the third condition was something that would be akin to their normal everyday map with everything on, right? The outcome measure was sentences and noise. And I should also note that this was double blinded because it's super easy for patients or participants to be biased in what they hear. And it's also really easy as an examiner to be biased when you're scoring things. So we made sure that everything was double blinded. So this is a really messy map to look at. The results aren't wowing in any way, but there is a trend in the direction that we would expect. So these are all the individual results here, but what I'll draw your attention to is just sort of these average results. Um, and this is, I should also point out, down is better on this graph. So it's a signal noise ratio. So the higher the bar means poor performance and the lower the bar means better performance. So if you just look at the average results here, the map that only had the worst electrodes activated, I guess, was the one that performed the worst. What's not so exciting, and it's a very small effect, it's significant, but whether that's clinically significant, I wouldn't guess, probably not. But it's sort of just this proof of concept that this is possible, that we can do this and actually modify the input that we're giving the brain. What's not super exciting is that these electrodes, the map that they had no electrodes activated or deactivated, excuse me, was really no better than when we selected the best electrodes. So what does that tell you? Maybe the brain's just smart and figures it out anyway. I don't know. Again, it was sort of like, okay, well, maybe we can modify it. And just like anything else in implants, everything's so individual. It's so hard to look at things at a group level. There's some of these individuals here down at this end of the graph that perform just as you would expect them. I paid them a little bit more to participate. Maybe that's why they did, but that's a joke. I didn't do that. But there's people that kind of fall in line with that prediction and others that are the exact opposite. But at the end of the day, this is what we shake out with. So there's some promise there is the whole point. Okay, so what else can we do? So I'm talking to, I think, just a room full of surgeons at this point, so I'm not going to tell you too much how to do your job, but there is some existing literature here to support maybe why we should be trying to preserve cochlear structures, right? So we already talked about the importance of the auditory nerve, and I really do believe in that. So do I think it's the golden ticket for everybody? No, but when we're talking about sort of these improvements in technology and improvements in performance, I always say that it doesn't happen. There's not one big thing that makes a difference, right? It's a sum of all parts. And so if we can improve If we can preserve hearing and, get 5% because of that, and another 5% from doing something else, another 5% from something else, well, great, that's going to add up to a lot, right? And so I think that, hearing and structure preservation during implant surgery actually is important. I do. Not for everybody, but when it's possible, I think it's important. So there's, it's hypothesized, I guess I should say, that a soft surgical approach would really help to minimize cochlear damage and really of those cochlear structures. So there's not much evidence on this, but there is a temporal bone study that evaluated 19 temporal bones of cochlear implanted ears. They found that whenever the electrode translocates, it causes this fibrous tissue, and that regions with extra fibrous tissue also had lower SGN counts. And this is not the only study that's shown this. They also showed poor performance growth in patients with translocations. And so I think a lot of times people, this translocation stuff and scalar location has been out there for a long time, right? Like 20 plus years from early studies that came out of WashU. But I think, a lot of people thought, well, it was just because it translocated. Well, maybe, but maybe it's also sort of damaging, the condition of the cochlea in some way and damaging the neural structures, which is actually affecting how somebody hears with their implant. Um, we know that scalar location is affected by surgical approach as well. And so this was just a review study done by Brendan O'Connell. And we can see if we use round window, cochleostomy, you guys are all familiar with these, but basically we know that if the electrode actually stays in the scala tympani, or sorry, different surgical approaches result more in the electrode staying in the scala tympani. And if it stays in the scala tympani, maybe that would help to preserve the neural structures that are there, right? The fact is some patients may come into a surgery without a whole lot of neurons left, but we have no control over that. But, when they do, maybe it would be better to, sort of do these things. But again, I'm not a surgeon, so I can't tell you how to do your job. So, but that's it. All right. And lastly, I'm just going to touch upon this. I know very little about this, but it is definitely something that researchers and some of the cochlear implant companies have also looked at, is either using a neurotrophin or using some sort of agent to actually protect the ear during cochlear implant surgery. And so we actually did a little bit of this. Yoash Raphael did a little bit of this work in combination with our lab at Michigan, where he basically induced a few different BDNF and a couple others, these ear neurotrophins, which is basically what it would just do is sort of protect the cochlea from damage. And in some cases, it would actually help to sort of regenerate these peripheral processes, which is what you're seeing here. So, maybe futuristic, there'd be a combination of potentially implanting, but then also giving some sort of some treatment up front to try to induce survival of the residual cells that are there. But I know nothing about this, so I'm not the person to ask about this. Okay, just to say that it exists and acknowledge it. Okay, so what have we learned so far? We've learned that the condition of the cochlea matters. It's important for how people hear with an implant. We've looked at some peripheral neural health measures that we can do that are sort of non-invasive. They're things we do in the clinic already. They're just doing it in a different way. And we can use those measures maybe to improve speech understanding, maybe. But what about the brain, right? So the brain, I will always say, is way more important than the ear. And so when I came here, I said, great. Yes, we know a lot about the way that the ear influences and the neural structures influence how somebody hears with an implant. But I was really curious to sort of look at things from the other way and understand a little bit better how the cortex encodes this information as well, and really interested in how cochlear neural health sort of interacts with cortical representation for the patients that we work with. So that's a lot of what I've done with research here and with mentors here. So I'm going to talk a lot about some temporal coding for a second, and specifically temporal pulse rate in cochlear implant processing. So I'll try to explain it for those who may or may not know this. But for cochlear implant processing, temporal fine structure is completely thrown out as we know it, and it's really replaced with this biphasic pulse train that is fixed in rate across all the electrodes. So not all of them. So Medell's processing strategy is a little bit different, but by and large part, it's just this random pulse rate. With some companies, it's a fixed pulse rate. It never changes. Another company, it changes based on compliance and impedance of the electrodes. And, it tries to get it as high as it can unless it has to drop it due to those factors. So it's all over the place is the whole point. And, it is one of those factors that we can definitely modify. So I can go into somebody's map and I can change it from 500 to 2000. The problem is we have zero idea what that does. And there's zero way to know. Sometimes people say it's better. Sometimes people say it's worse. We really don't understand anything about pulse rate or what we can be doing as clinicians to perhaps make things better for people. Is that something we can modify to improve performance? No idea. So that's really one of the things that I wanted to focus on, again, coming from a clinician standpoint, is looking at temporal cues and pulse rate. Temporal cues are pretty broad, I realize that, but they're super important for speech understanding, and they're particularly important in older brains and older populations, which, if you look at our average adult clinical population for adult CI recipients, more than half of them are 65 and older, all right? So there is they should always look at aging, because aging is always going to play a role in sort of what we look at and how we look at it. So temporal fine structure is replaced with biphasic pulse train. Again, it's most often fixed in rate, and this interaction between pulse train or pulse rate and performance is super unclear. Based on animal studies and a few human studies, what we know is that increasing the pulse rate might improve coding of speech cues. So there's some evidence to show that it improves coding of silent temporal gaps, which is super important. And that's what we're going to focus on for the rest of the slide. There's also for the rest of the talk, there's also some evidence to suggest that increasing the pulse rate might actually worsen encoding of speech cues. So there's some work that's been done that has looked at amplitude modulation, for example, and that it sometimes gets worse as you increase the pulse rate. And then, of course, it might interact with chronological age, but we just don't know any of these things. We're going to focus for the remainder of the talk on really the silent gap perception and encoding of that at the level of the auditory cortex. So temporal cues are super important for speech, but they're altered in CI users. We're going to focus on silent gap detection primarily because it has shown aging effects. There's been a little bit of evidence that has suggested maybe it interacts with pulse rate in some way, although we don't really understand it. And then also because it's really, really important for speech understanding from a very phonemic level to more of a sentence structure level, right? So in order to really hear speech clearly and to segment things, you have to be able to have, sort of this keen perception of just gaps in speech, or else it all gets mushed together and it's no good, right? So we're going to focus more on silent gap detection for these next experiments and how it interferes or interacts, with carrier pulse rate. So our first prediction is that encoding of temporal gaps is actually improved when using a higher compared to lower pulse rate. So this would be consistent with some of the existing literature that's out there. And our prediction number 2, though, is that this effect is actually modulated by peripheral neural density. So we actually see more of an improvement in cortical encoding of gap detection when we increase the pulse rate, but more so if we're stimulating a portion of the auditory nerve that has robust population of neuron fibers, right? So the idea is that if there's more of a neural substrate there at a very basic level, it really helps to code that signal a little bit further up, and it primes the cortex a little bit more for encoding of that information. Okay, so we actually have quite a few more people than this enrolled, but again, still sort of working through the data. It takes a long time to process this data, but so far I'm going to show you results from 14 adults. They're all experienced CI users. All stimuli were loudness balanced, which is super important. I point that out only because if they're not, it can really, really affect the cortical responses that I'll explain to you. We used direct stimulus research interface. So this is all Cochlear recipients. There's a research interface called NIC that they offer, and it allows you to stimulate electrode 5 with whatever stimulus parameters you want. So you can sort of very precisely control that. And we used a within subject design, as I'll explain. So first thing that we did was we measured the ECAP measures that we talked about at the very beginning on each electrode for these two interphase gap values, so we could calculate that interphase gap effect, which again, going back to the beginning of the talk, we know tells us about the number of neurons that are there at one electrode relative to another electrode in the ear. So these are just some examples of these AGF functions for one participant, one ear going from base to apex. And then the next thing that we do is we actually fit each one with a linear slope, and then we calculate the IPG effect for that slope. So for each individual, we found the electrode with the highest and the lowest linear slope, which again represents better and poorer SGN density respectively. And so for this individual, electrode 18 had the steepest slope. Here, we would hypothesize that that has, relatively speaking, the most robust population of neurons. Adjacent to that, electrode 9, we would argue would have sort of the lowest number of fibers remaining. So these two electrodes were used for all EEG and psychophysical silent gap detection tasks for each individual. So we just use the same two electrodes for each individual over and over again. So this is the EEG responses recorded with a 64-channel cap. The task was simply this. So it's just a pulse train, and it is every so often there is a very brief silent gap. We have 9, 12, or 15. Don't worry about that. We tried out a couple different things to see what would work and what would elicit a response. But basically, this is just an offset response. So it's the electrically evoked, some people call it the acoustic change complex. Here it's not really acoustic, so we're using auditory, but basically it's a cortical evoked response that you get just by a change in the response. So in this case, it's an offset response. So when this pulse train stops, the cortex responds because it notices that there's a change in what it's hearing. Otherwise, it just sounds like a continuous signal or a continuous beep to people. We used those two pulse rates. We used a very low pulse rate of 500 and a higher pulse rate of 3,500. And then we stimulated, like I said, two electrodes for each ear, one that was estimated to have a higher and lower density of fibers or higher and lower ECAP linear slope. And then you do run into quite a bit of artifact, obviously, RF artifact, among other things, when you're doing these types of experiments. But we used ICA in order to identify and reject artifacts, and it actually works really well. It works better than what we thought it would. So this is just an example of the cortical responses that we obtained from one patient. So here, these are the responses with the 500 pulses per second. This is with 3,500 pulses per second. And one of the things that you can see already is that the response for the 3,500 pulses per second is a lot bigger than the 500. So that's pretty obvious without looking at anything else. The other thing that we see is that, again, this is the electrode 9, sort of the poor performer, and electrode 18 has a more robust population of fibers. And you can see here that there's, for the 500 pulses per second, there's just a little bit of a difference here. And for the 3,500, there's a bigger difference between those two electrodes. So far, based on our one person, this fits our hypothesis really, really well. So we should have just stopped the study there, right? But if we want to look at the average responses, we actually see a similar trend in the data. So we see that, again, if we just look within 500 pulses per second, we find that there's larger N1, these are, again, cortical responses, larger N1 amplitudes on electrodes with higher ECAP slopes. So basically what this means is that you get larger cortical responses when you stimulate an electrode in the auditory periphery that has a larger population of fibers compared to one that has a lower population of fibers. The N1 amplitude increases with increasing pulse rate. So even if you sort of stay on the same electrode and you just increase the pulse rate, that N1 amplitude response increases. And we also see that there's a bit of an interaction between these two variables as well. So basically what this looks like is that the gap between here is a little bit smaller than the gap between here. But there seems to be this interaction between these two variables, meaning that this effect of pulse rate is greater when stimulating electrodes estimate to activate a higher density of peripheral neurons. So again, there's this interaction between those two variables, okay? What's also interesting, and this is data that we're still sort of getting into, is that the silent gap encoding with a faster pulse rate, so this is, I forgot to put this on here, I apologize, but it's basically this condition, it's 3,500 pulses per second. If we look at the amplitude response in the cortex, we actually find that with it actually happens with a slower pulse rate too, but these cortical amplitudes are also significantly related to how people understand speech and noise. So this would be a larger amplitude here. Those individuals perform better in sentences and noise. And these individuals here with a smaller amplitude, this N1 amplitude, perform poorer in noise. So there does seem to be, even aside from, stimulating good and bad electrodes in a way, there does seem to be definitely this relationship between how somebody hears speech and what their cortical representation of speech actually looks like using an electrophysiologic measure, right? Okay, we have a couple minutes. I'll go over this. So this is just a parallel psychophysical gap detection. So instead of having them wear a cap, they're basically sitting down in front of a computer and doing boring psychophysics, which basically involves, hearing a couple beeps and clicking on the thing that's different. So in this case, they'd hear beep, 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 and they would have to click on this one because they should be able to hear that gap there that's not there in the other three. And it's important to understand sometimes if the behavioral perception actually matches the cortical representation of that. Sometimes it does, sometimes it doesn't. In this case, just to sort of skip through a couple things, we actually find that it's a pretty similar pattern to what we observe with the EEG data as well. So in review and discussion, cochlear neural health, really neural survival, and more importantly, I think synchrony, even though I didn't talk a lot about it today, is an important factor that influences CI outcomes. Again, as an audiologist, I think it's exciting. Again, we don't have a lot of things that we can do when we're sitting in front of a patient and say, okay, we're going to think outside the box on this one. And instead of just doing the standard settings, we're going to do a couple extra measures and modify these things and see if it actually makes a difference. And so maybe that's electrode deactivation. Maybe it's changing the pulse rate. I think we need to understand that a little bit more. But it would be wonderful if we had a few more tools in our belt in order to improve performance when things just don't go as well as what we had planned. We also have some other projects that are overlapping with these that examine interactions with audiovisual speech. So that stuff is super interesting. There's big aging effects in audiovisual speech integration. A lot of times from what we're seeing, the results in our cochlear implant patients mirror that of those with normal hearing when it comes to AV integration. But there's some interesting things there. And I don't know, maybe in a couple of years, I'll share that with you if I get invited to do this. But it's a whole other talk. It's a whole other thing, but there's some really cool stuff with AV coming out. And then again, all of this stuff really has really strong aging undertones to it that I didn't even touch upon today. And so we're looking a little bit more at how aging really modulates all of these findings that I discussed today. So with that, I'd like to acknowledge collaborators, consultants. Again, some are here at MUSC, some are previously some of my colleagues at the University of Michigan, and then our funding sources as well. And that's about it.
That was an awesome talk. Thank you so much for spending the time and speaking to us. I have a few questions myself, and then I'll also open it up to others. But I was really interested in, and I know you didn't speak that much about the synchrony topic. And it sounds like it's something that you continue to study and be interested in. And I was wondering, do you think that, and by that I mean synchrony at the level of the like auditory nerve firing, not like gross neural synchrony in the cortex, but
although we
do
are
you
looking
think
at that too. I just don't have, I'm super interested in just sort of cortical synchrony as well. And there's other measures that we can look at for that. But anyway.
Do you think that from a stimulation kind of coding selection electrode activity perspective, you could modulate synchrony at the level of the auditory nerve? And do you think the second part to that is like, do you think the ability to modulate synchrony or even the impact of synchrony, it would be dependent upon the sort of overall neural health of the nerve, like how many actual fibers are left, like that can fire together versus like 100,000 versus 10,000, or do you think that that would have an impact?
Yeah, so first question, I would say that that's sort of so hoping to look at that, and I'm in the process of writing my next grant, which is due this summer, and that's one of the things that we're hoping to really focus in on a little bit more is just synchrony. And if you can modulate, for example, pulse rate is a great example. Theoretically, if you slow it down, there should be more synchrony of those fibers. It gives more of the auditory neurons a chance to recover and then ready to fire again, right? And so it's always been one of those suspicions with these really super fast pulse rates. It just doesn't always make sense to me based on what we know of the auditory nerve firing patterns and how that's reflected higher up. So, I do think that if you increase the pulse rate, it should create a more synchronous firing pattern or more opportunity for that, right? But we don't know that for sure. Your other question, say it again. Sorry, I had it in
Kind of
my
like
mind.
the interaction between like the
Oh yeah,
number of available neurons
yeah.
and the impact of synchrony.
So from what we're seeing so far, no. It's actually interesting, but no. And I should also mention that the synchrony is thought to really be determined by myelin. And that's, again, this is nothing that we've actually measured in SCI patients, but it's just from normal hearing or normal hearing aging literature. And they see that it doesn't relate in that population either. So, you can have a big response. It doesn't necessarily mean that it's a synchronous one. We do see some effects of uh, just just the number of fibers that are remaining. We do see that that goes down with age, for example. Um, and so, sure, like I think that they're somewhat related, but they're not. There's very clear evidence that they can also be two very different things as well. So we're doing some parallel experiments, I guess, in CI to sort of try to help figure that out. Exactly. Yeah.
Well, thanks. And the second question I had was a little less well formed, but it got me thinking because I mean, an interest of mine is in tinnitus, neuroscience of tinnitus, and sort of implant related suppression of tinnitus. And I was wondering and I was thinking about it because you're talking about the silent gap detection in humans. And when I did my PhD and looked at mouse models of tinnitus, that was like this kind of flawed but main behavioral model we'd
The
use in
startle
mice, this
response. Yeah.
startle response and impaired
Yeah.
gap detection.
Yep.
And I was wondering if you thought that, I mean, we think about tinnitus being in the brain, but obviously electrical stimulation of the ear and the brain suppresses tinnitus, um, particularly in CI patients. Do you think that your measures of sort of peripheral neural health or peripheral neural synchrony, like, do you think that that might have an impact on on the degree to which an implant would suppress or not suppress a patient's tinnitus? Have you found that, or would you have any reason to believe that that would be the case?
No idea. I don't really know. Yeah, I'm not sure. I mean, it's a great question, but I don't know for sure. Yeah.
I want to give anyone else a chance. I also I have recorded this, and I know that 5 o'clock is hard on Mondays for some people, so I'll definitely be sharing it online as well. But before we thank Dr. Leyzac and let her get on with her evening, does anyone else have any questions this evening? Okay, Dr. McCracken is raised his hand. I think you can speak, Dr. McCracken. I don't think I need to unmute you.
Yes. Yeah, can you hear me?
Yep.
I just lost. I don't know where my screen went. Okay, there you go. So question, ECAP measures kind of measured at the time of implantation, to within subjects, like kind of a year later or so. I mean, have you guys done that a lot and see if kind of the changes that happen as a result from implantation?
I have not. They were starting to get into that right when I was leaving Michigan. They definitely change a lot intraoperatively, but a lot of that we don't exactly understand why. So some of the animal models suggest that right after cochlear implantation, you see sort of a decrease over the next in the slopes and the amplitudes, right? So you see this decrease in the ECAT measures and the super threshold ECAT measures, and then within a week or two, they're like higher than they were the day of surgery. So there's clearly some sort of like recovery that's going on that I don't think we quite understand. And there's also, you have to think about it too. There's just like the impedances and all of those things, all of that's way different intraoperatively. But once they're stable, they seem to be stable. So yes, we do see these changes. Of course, they can happen. But I think there's a study that was done many years ago by Chris Brown that has looked at this. It's been a number of years, but it's really the only one to look at very long term, like several years down the road for these super threshold measures. And what they actually found is that they're generally stable, but there's not a lot of evidence. I don't know. I would say yes, but there's also not like 50 studies out there that have looked at it. So.
Great. Great talk, Kara.
I thought you were going to ask why I didn't look at the CIQOL, but
that's an offline question.
Well, anyways, that was a really great talk. Thank you so much, and look forward to seeing everyone at the next talk. And I'll get this talk uploaded shortly. But thanks everyone for being here. Thanks so much, Dr. Leyzac, for taking the time to talk to us, and have a great night.
All right. Thanks, everybody. Bye.